Okay, so this is going to be our tutorial on GimmeMotifs for motif finding. So essentially, motifs are DNA or RNA sequences. These usually have important functions like altering gene expression. That's kind of what you're seeing in this figure on the right. So these are proteins that have been expressed from DNA, and they can bind to DNA, and that can alter protein expression. And proteins can also bind to like cell receptors, which can also impact gene regulation. A lot of the work for looking at motifs was done in the 1980s, and that's when you first see figures like this, which are called motif logos. They're kind of showing for a single motif how much, almost the fraction of them that have, say, T in the first position, G in the second position. And there's a lot of tools to be able to do this. We're specifically going to be looking at one in Python called GimmeMotifs that has been used in the past to analyze motifs.
There are a few necessary things that need to be done to get GimmeMotifs installed into your computer so that you can begin using it in your Jupyter notebooks. And the first thing that you should do is start by opening a new terminal window and then use the conda method for package installation. This is generally recommended because it's easier than using pip install as opposed to using just the conda method. And so by doing this, first you would type into your terminal conda create -n motif python equals 3.8. Then this would be followed by activating the environment by saying conda activate motif. And then you would type in conda install -c bioconda GimmeMotifs. And then you would type in gimme-help so that you can confirm that the actual tool is installed in your environment. The next step that you would take is to configure the package, and so this would be done by typing into your terminal gimme config. And then with this can be followed by doing a test installation by typing in the following. What this is important for is to verify that you're actually able to take some of the installation packages from the actual GimmeMotifs program so that you could verify that, one, you actually have the package installed into your environment and that you can also get downloads so that you can begin to use tools. And so for this instance, you would install Jasper in order to begin coding so that you can begin to analyze mRNA sequences and do motif finding analysis. In some cases, you might have trouble trying to get this package to install onto your actual Python environment. And so in some cases, you may want to check what kind of Python version you have and what kind of compatibility it has with the package that you're trying to install. You should also be sure to look back and see whether or not you have all of the necessary dependencies that should be installed onto your environment. And if you have checked that, I would move on to consulting the actual documentation to look at specific errors that might arise when you're using this specific package. And so one of the things that has actually helped me when I was getting this package installed onto my own device was by constantly looking at the documentation and also consulting AI devices so that I could look at different solutions and type in different errors to find different fixes in order to be able to configure the correct kind of environment so that I could run the correct kernel in my Python environment when I was creating some Jupyter code. And I found that to be very useful as well.
So for GimmeMotifs, the math is primarily based on this equation, which can be used to calculate the weighted information content. So to to use this to compare two different motifs, we have our two motifs, x and y, and then two more equations, the information content and the differential information content, and then a constant c. This can be tuned to how much you want to punish differences between nucleotides in x and y. GimmeMotifs uses a constant of 2. And so then for information content, we have this value here, which is looking at the frequency of a nucleotide appearing in position I, or if you're looking at motif Y in position J. We'll go over an example where we talk about that in more detail. And then we have the frequency of background, which is essentially just 0.25, assuming that there is an equal probability of any of the nucleotides appearing in a position. That's not necessarily a true assumption, but it makes the math a lot simpler, and so that's an assumption people usually make for this. Then we have the differential information content, and so it's also taking an x and y as compared to information content, which just takes in one of them. And so essentially what this is looking at is its information content, but just the difference on a nucleotide basis. And so this will be larger if there is a bigger difference in the nucleotides in a position. So using these three equations, we'll be able to quantify the differences between x and y. In the case that you're not looking to compare two motifs, all you need is information content, and this is what people use to make the logos for motifs, which will show you how much of each nucleotide appears. So I'm going to go over an example of going through this math with two motifs. We have x here and then y here. We'll be using the first column as position 0 and then the last column as position 3. And just looking at this here, you can see there's really good agreement in the first two columns with A and C. And then the third column, or position 2, there's a big difference between them, and then in the last one they're also the same. So then we'll go through calculating the frequency so you guys can see how these change with different nucleotides. So with the in position 0 in the first column, it's all A, and so what you can see is frequency is 1. For C, G, and T, the frequency is going to be equal to 0. And so then looking in position 1, it's the same thing, where the frequency of C is 1, frequency of A, G, and T are equal to 0. And then in the next position, we have frequency of G equal to 1, and C, G, and T, or sorry, C, A, and T are all equal to 0. And then the last position, or position 3, which is the one that I made interesting, has A and T in it, and so then the frequency of A is 1/3 because 1 out of 3 of them are A, And then for T in position 3, we end up with 2/3 because 2 of them are T. And then G and C are equal to 0. So then we'll be able to calculate information content using these frequencies. So for positions 1 through 2, this is pretty easy because the frequencies are all 1 for A, C, and G, and 0 for the rest. So information content ends up being 2, which is the max value of information content. And then moving on to position 3, which is a little more interesting, you can see that the frequency of A has been plugged in here, and the frequency of A has been plugged into the log as well, and then added to the frequency of T times the same thing where the frequency has been plugged into the log. And then if we wanted to make one of those logos, we could take these individual information content values for the different nucleotides and plot the letters and proportionally to the size of the information content. So then moving on with this example, we'll continue using the same motifs that we were using in the last slide, but we're going to continue on and calculate the differential information content for X and Y, and then use that to calculate the weighted information content. We're not going to go through all the positions just because it's a little redundant to do all of them, but we will go through the 0th position, which will show us what the weighted information content looks like when all of the nucleotides match, and then we'll go through the 2nd position here, position 2, where X has all of the values matching with G, and then Y has ATA. And that'll show us what happens to weighted information content when they don't match. So looking at that first position for the nucleotide matching example, so essentially we just need to plug in the 0th position for X into information content, and 0th position for Y into the information content, and then both of them for the differential information content. So similar to the last example, the information content is just going to be 2. All the nucleotides match, so we're getting the max value of information content. That also makes the differential information content pretty easy because all the nucleotides match is just going to be 0. And so then the DIC term is going to go away, so we're not going to see what that does. The information contents are equal, so we multiply them, take the square root, and we end up with 2. And so this is the max value of weighted information content, which is indicating a really high correlation in the 0th position between X and Y. Yeah, and something to note for this is that in order to look at a comparison between 2 motifs, you need to align them first. Otherwise, this isn't going to be as meaningful if you're just comparing positions. So now we're going to move over to position 2, comparing X with the 3 Gs and then ATA and Y. So same deal, we're going to be plugging X2, Y2 into the weighted information content equation. Information content for X2 is going to be 2 because all of them match, and then for the Y2 information content is going to end up being 1.08. This is what we saw in the last example. That one was ATA, but because you're summing all the information contents from different nucleotides together, it doesn't really matter how much, or which nucleotide's bigger for information content. Information content's just telling you how much above the background a nucleotide's appearing. So in the case of 1.08, we have 2/3 of our nucleotides are A, and so that's quite a bit above the threshold of 0.25. But naturally, if you have a log base 2 of the frequency over 0.25, if your frequency is less than 0.25, you're going to end up with a negative information content, which is just indicating that it's appearing less than the background frequency. And so then we can move on to information, or the differential information content. This one's a little more tricky to calculate. So essentially, we'll look at A, plug in the nucleotide A and our position as well. Calculate information content for both of them, look at the difference, take the absolute value, and then sum all of them. So here, this is for A for both of them. So if you think about the X motif, there's 0A appearing, so the frequency here is going to be 0. So essentially this is just the Y, and that's where you get that 2/3 here from. Then if you move to T, you see the same thing, except it's switched to 1/3 because 1/3 of them are T's. And then the last one is just log 1 over 0.25, and that's representing the G's, right? So because there's no G's in Y, it's just going to be the max value. So then we can plug this into the weighted information content equation. So what we can see is that because they didn't share any nucleotides in this position, the DIC value ended up being kind of big, and so even though both of them had high information content, it didn't end up being a positive WIC value because the nucleotides that they had in that position were different. And so what the WIC ends up telling us is how well the nucleotides in that position match between X and Y.
So now that we've gone over some of the mathematical basis for which motif finding works under, we can begin to get into some of the practical application of why this is important and where this can be applied. And so one case of where this can be used by researchers is by analyzing heat shock proteins, otherwise known as HSPs. And so what this is a family of proteins, and they're released in specific biological conditions when there's exposure to some kind of stressful event. And so what this can include is being exposed to high temperatures or extremely low temperatures, having exposure to toxins, or even being exposed to oxidative stress. And so where GimmeMotifs can be used is to observe some of the conserved motif structures within these mRNA sequences, such as the heat shock element, which is essential for looking at HSP expression. And so where this can be further used is by assisting researchers with being able to understand better how certain cells can respond to temperature-related stresses by the upregulation of HSPs. And so you might ask yourselves, why would you want to use GimmeMotifs and motif finding in this way specifically, where you could potentially use other kinds of tools to do this type of analysis. And so one of the main things that you could also consider is by looking at efficient motif discovery by using GimmeMotifs in this way. And so what this allows you to do is to quickly identify motifs that exist within large genomic datasets. And so it'll save you a lot of time during experimental research. And with this, there's also a high degree of sensitivity that comes into play where you can detect low frequency motifs that could also have some kind of significant regulatory function in a certain stress related response. And to wrap that up, this is also a very customizable method. And so what I mean by this is that this method of motif finding can be adapted to different kinds of organisms, or even by looking at different stress conditions. Like we mentioned before, it doesn't necessarily have to be temperature related, but it makes it so that using this tool is a very versatile tool for looking at various research projects and being able to manipulate different parameters so you can look at an array of different outcomes. Now that we've established the basis for why we would want to use GimmeMotifs in a practical application setting and use it in order to look at a certain subset of species in order to understand how they behave under a certain set of conditions, I want to emphasize why it is important to look at all of this within the context of coding and using a program such as Python. And this is really because it helps for researchers in particular to look at doing analysis without necessarily having to go out and do a time-consuming or computationally expensive experiment. So rather than doing the analysis in real time, you could also simulate these things in a program such as this so that you can kind of guide your research in a more effective manner. And so with all of this being said, what I have for all of you is a Python script that my group and I came up with together so that we can visualize a specific genomic motif that repeats in a certain subset of gene sequences that we acquired from NCBI. And so what we're looking at in particular is the motif GAA-X that repeats in HSPs. And so that X at the end of that little sequence is some kind of nucleotide. It could represent any kind of nucleotide. And what we aim is to look at 7 human mRNA sequences in a FASTA file format so that we can continue to do our analysis on these. And so I also want to mention that we have our kernel set to the right kernel. This is important to note considering it'll allow us to use some of the functions within GimmeMotifs and any kind of other analysis techniques that we would be interested in order to do our analysis. This could be used for a different array of reasons that a researcher might be interested in different aspects of whatever it is that they are analyzing. And so with this, we also have our libraries and our setup. And for good practice, it's always important to make sure that you have the right kinds of files that are uploaded, so that way you can also run certain functions to be able to perform your analysis effectively and properly. And so, for example, we have RE that's imported for regular expressions, bio.seq.io from BioPython so that we could read the FASTA files that we have. We also imported the logo maker so that we could create the sequence logo for later. We also have matplotlib so that we can make plots, and we also created a file path as well in this code so that we could download and look at our files for later in our code. And so with this, now that we have everything sort of loaded into a list that could be analyzed further, the next thing that we also set up is by looking at the actual motif finding. And so with this, what we first did is we defined the motif pattern to be GAA, and then we kind of just used a regular expression to do so. And the period here means that any one nucleotide, and so this captures any sequence that starts with the GAA and then followed by any kind of base. And so for each file, we loaded the sequence, we converted it to uppercase to avoid case mismatch, and then we used re.findIter to locate every match. And so we extracted each 4 base motif pair, and we stored it inside of a list called sequences with motif. And so after we did this, we found what all of the positions were of that nucleotide sequence that we were interested in. And then we found the lengths of these mRNA sequences as well so that we could create our heat map and be able to perform a subsequent analysis by looking at what the output positions are of like where the gene is in relation to the motif density and also relation to the nucleotide position to just get an overall understanding of where this motif lies in with each of the mRNA sequences and how they compare to each other. And so following this, we also created a logo making code where, if we have any motifs that we found, we would try to convert the motifs into a count matrix, which is important because it helps to tell us how often each of the bases appear at each of the different positions. And then we were able to normalize each of the rows so that we would be able to reflect the frequency of each nucleotide at each position within each of the mRNA sequences' respective positions. And so with this, we also plotted the logo and we were able to continue to just visualize certain spots, such as, where some of these nucleotides would be located. And so what we really see is what we'd expect, where the first three positions will almost always be G, A, and A. And then the fourth position tends to vary from this graph. And so what we can see is that looking at ATG or C is that there is some relative abundance in each of those respective positions where they appear on the graph. And this is important because it helps us to be able to understand better um, the potential variability in the motif's recognition or function. And so with all of this, there's a lot of ways in which this script could also be modified or extended, where we could search for longer motif patterns or different patterns in general just within the mRNA sequence. And we could also get some kind of output to match the sequences to a file for further analysis. And we could also transform the count matrix into an information logo, which would help us to highlight which positions are also the most conserved. And so in summary, what we were able to show by having the script is that you can use Python in tandem with GimmeMotifs to be able to scan genomic DNA sequences so that you could look for motifs, visualize their variability across samples. And it's a small but effective example of how you can use a computational tool in order to help support further biological discovery.
So for the results, we first have a motif frequency logo. So this just shows the nucleotides that are conserved in the heat shock motif. So for this figure, we first have, in the y-axis, we have frequency, which is just how likely, I suppose, it's there, that is there. And the position is just the position in the motif. So what we have here is nucleotides. So in the very first position, which is on the very left, we have G. We only have G. That means that we can confirm that this is a G. And then at the second and third position, we have A's. So we can definitely confirm that GAA is highly conserved in the first three positions after the heat shock. And then at the fourth position, it could be any one of the nucleotides. So that's why we would label it as X, so GAAX. So this is a pattern that's actually common and well known among the heat shock elements. It's where the heat shock transcription factor tends to bind during a stress response. And this just confirms the ability of the tool to detect conserved components correctly. Okay, so for the second result, we have a motif density heat map, which just shows where the motifs are for each of the genes. And so the x-axis is the nucleotide position, and the y-axis is the specific genes. And we're looking at, I suppose, 7 different genes, or probably just 6, because the 0 might be some sort of control, but we still see 0 to 6 genes. And the vertical lines that you see of varying colors are the actual motifs. So if you can see on the right, we have a gradient, which is why we consider this a heat map. And some of the motifs are on the lighter side or the darker side. And the darker they are, the more motifs that are present there. And as you can see, most of the motifs are around 200 bases to 1500 bases in the position, which is very important because that's around where the promoter region is. And that's also another important thing. This is also where we expect the transcription factors to bind to regulate gene activity. So this figure confirms that the motifs we found aren't just random, they show up in biologically relevant spots. So putting this all together, we can say that the tool GimmeMotifs effectively discovered the conserved GAA core motifs, and that these motifs were discovered around the promoter region, or where the transcription factors, the heat shock transcription factors, where they typically bind, the 200 to 1500 bases that are upstream of the start site. And of course that confirms that the heat shock transcription factors are binding to the heat shock element motifs, and when they bind, that regulates the genes. So all of this basically shows that the tool GimmeMotifs is effective at what it's supposed to do, at finding these patterns. But not only that, but it also gives us results in finding what is actually biologically relevant in this context of the heat shock biology. So to wrap everything up in the summary and conclusion, we can pretty easily deduce that the GimmeMotifs is really good at finding motifs for specifically our case, and really for a lot of other cases, but for our case, we deduced that it was really good at what it did. It found those heat shock element motifs and found them in the locations that are biologically relevant. And specifically looking at the GimmeMotifs tool, we have also deduced, as well as it's been proven by a lot of other research projects and publications, that the tool is very fast, it's robust, and it's very accurate at analyzing everything from tissues to conserved DNA motifs, which is what we did in this project. Now, it uses an ensemble-based approach to find these motifs, to find whatever it is, which basically the ensemble approach means that it's using a variety of different tools, multiple tools, to come up with the certain results, which that makes the results that much more reliable. And of course, the way it works is it looks for patterns. For our case, for the motifs, it looks for the motifs in the DNA sequence. For us, it was GAA, and it looks for them and sees if the patterns match. And for the logos, it did pretty good, the GAA, and we just had the fourth position being whatever it could be, but it was able to find them. And it can find these patterns, whatever it is, it can find them without prior knowledge, which is very useful for a lot of different applications. So moving on, the install process, it's pretty adaptable. You can do, there's many ways to do it, but for the ones that we know of, there's a command line interface way, which is just kind of in the command line or terminal or Anaconda, whatever. And there's also, you can install it through Python API. So, and this kind of makes it more versatile in the amount of projects that it can be used for. A lot of different researchers can use this for a lot of different things. And best of all, it's free and it's regularly updated. So I would say that this is highly recommended for people that have computational experience, that want something analyzed and want patterns recognized in this type of way. So before, we just talked about the positives, the many positives of the GimmeMotif tool, but I think it is beneficial to talk about the negatives as well. The first one being, it is very difficult to install. There's a lot of different codes, and it can really depend on your directories and your system. It's very picky on the commands that you need to implement. There you have to put in commands into a terminal to get it installed and get it working, even if you are just using the Python API. Now, one of the main reasons why it's so difficult to install is because there is no Windows, or I'm pretty sure even there's not Mac support either. It's only available on Linux to analyze the FASTA files. But the reason that we can use it is because there are methods to get around this, to use Ubuntu or any other method so we can utilize it, but it just takes some effort. And for large data sets, the tool requires an immense amount of computational power and resources and a lot of time to get effective results. So that's one of the biggest downsides of this tool. And since there's a lot of factors to consider when installing and when using it as well, it's not very good for beginners. There's just limited guidance on what to do at certain points, and the installation process can take a long time to master, even with the help of so many different tools online, people or AI. It could still be a full day's work. It could take hours or whatever to actually get the thing installed. And for people that don't have much of experience in coding in command lines, terminal, Anaconda, Python, whatever, it could be very difficult to install. So here we're trying to link basically the GimmeMotifs tool and the results that it got. For the results, we just talked about what was happening, but here we're just connecting the tool as well. So all in all, just looking at both of these figures, they're very easy to understand. We can see on the very first figure that the GimmeMotifs tool found the nucleotides in the appropriate positions in the motif, and that's found by looking at the patterns. And for the second figure, the tool was used to confirm the motif's position within that 150 base position or distance, or whatever you want to call it. And of course that was relevant because it was around the promoter region, which is where the heat shock transcription factors would bind to the heat shock elements. So that made it biologically relevant. So all in all, these visuals show how practical the GimmeMotifs tool is at studying the different kinds of gene regulation responses. Okay, so to wrap everything up in the conclusion, we can say that the GimmeMotifs tool is a very powerful tool for finding motifs in DNA sequences. Just based on our study, the other studies have shown how good it is at other things like tissues and stuff like that. But just a little bit of an observation, it could definitely be improved to include different cross-platform, like Windows, Mac, whatever. And just because it's very hard to install, so an easier install process, some guidance would do a lot in terms of making this tool a lot more accessible to people with less experience. And not everyone has Linux. A lot of people have Windows and Mac. So I think getting over that requirement to use Ubuntu or whatever would be a tremendous thing for this tool. It would be a good thing to remove that barrier. And how it uses multiple tools makes it, to get the results, makes it valuable for studies involving stress response genes, as we have confirmed in this project. But with people that may not have as much computational experience, like biologists, they may not have the necessary experience to work with something like this. And if they require motif analysis, they may have to find another tool than using this one. To wrap this all up, though, for the future direction, this may include applying the GimmeMotifs tool to other stress response genes, since we know that it's really good at finding those conserved motifs or whatever we're trying to look for. We're looking for patterns, basically. That's what this tool is really used for. So definitely looking at other stress response genes, because we're just trying to find something, what is conserved, what is relevant, to make sure that it connects, that type of thing. So I think that's very important. Thank you. So to wrap everything up, we want to look at some open questions. And the first one would be, how would the results change if a different database were used? So different database might give different motifs, which could change our biological conclusions. And for number 2, what is the biological relevance of the motifs found? So, we found our GA motifs, but we should verify if they're functionally active binding sites, right? And for number 3, are they known or unknown transcription binding sites? And that's kind of like, it's unclear whether they are already established sites or possibly novel ones. And for number 4, how would this tool perform in RNA motifs or non-coding regions, on RNA motifs or non-coding regions, excuse me. So it could be interesting to test the tool beyond just the promoter region, and I think that would be an interesting answer, I suppose. I'm just looking into that. And then the final question is, could the ensemble method miss rare but important motifs? So using the multiple tools to look for patterns, it helps the reliability, yes, but it might overlook low frequency patterns. So I think another weakness for this method, this tool, is that it doesn't do well with noisy data or patterns that are less frequent and stuff like that. So these are the type of questions that we would have for our open questions section. Thanks.